Welcome back to the another series of the lecture, which is about the targeted docking, which is also called a site-specific docking. For this purpose, we need to know about the residues of the binding pocket, which can be know by the literature or by finding it using different software, using CASTp or the Discovery Studio or Chimera. Today, we are learn how to find the residues of the binding pocket by the Discovery Studio. We are open our protein in the Discovery Studio, converted to mouse mode, so we can rotate our protein in the 3D view. You can see there is two ligands are attached there. To find the binding pocket, just simply click ligand interaction. So you can see the ligand with these lines or the interaction by moving it in the 3D view. It's the clear structure, and click on show 2D diagram. It is the 2D diagram of the molecule, which we can see on the screen. It tells us about the different residue which interact with the ligand within our protein. Okay, it's we need to write it on our register. Okay, go on tools, click on view interactions, and again the ligand interaction to see it in the 3D view, to find residues in the 3D view. Simply right click, label, add, and click here the amino acids and find the ID and this 3D letter, so we can know about the name of the residue, interacting residue. Okay, there. Simply apply OK or click. Now you can see easily see the interactive residue of proteins with the ligand. Write it on your register, these residues. It helps to perform the site-specific docking. Okay, simply we show it in the with white background. Click receptor, so you can see its interaction with the receptor. Okay, unclick the receptor, the receptor will unappear. This picture can be used in your thesis or the research paper. Simply we can do it for knowing about the residues of the binding pocket. This procedure is helpful in performing the site-specific docking. Okay, if you are know that this is your interesting binding site and there is a ligand attached there, then you can find the residues by applying this procedure. If you are not know binding pocket or the binding pocket residue, there's another method of CASTp which is shown in the another video, how to find the binding pocket of the protein and to find the interactive residues of that protein. So we simply open Chimera and load your macromolecule, which is in protein. Click on AutoDock, make macromolecule. Initial step is simple to the blind docking. There is only the difference in making the grid. Otherwise, all steps are same as in the blind docking. Simply click icon in the open babel and load the molecules or ligands one by one. Okay. We input 3 different ligands, right click and minimize all and convert all to the PDB-QT format. Now you can see the ligands converted into PDB-QT format and the molecule also converted in the PDB-QMAT. Go to the Vena wizard, click start, and you will have to double check your ligands and the proteins will be selected. Simply click the first ligand and press shift and last, and then all will be selected. Here is the difference for selecting making a grid. Okay, you simply click the plus icon. Now you show the amino acids of the first chain. You have to select those residues which we were noted on our register by seeing the 2D or 3D structure of the protein in the discovery studio. Select only those residue leucine 17 as selected. Hmm. Simply right click display label atom. This method repeated again and again until all the residue will be selected. Okay, right click display label atom. This is the method which is only done by doing the site specific or the targeted docking. This step is skipped in blind docking. Okay, now you maximize the window and you see the white written up residues in the grid box. Okay, we set up our grid around these residues. None of the residues is outside of the grid. All the residues are highlighted here is within this grid. Okay, grid is not too small or too large. It is the crucial step for performing it. Be careful while setting a grid. Um, uh, Watch, see it or check it from every side that none of the molecule will be outside, should be outside of the grid. Okay, check it again and again. It is very crucial, and if it's not done accurately, then results will be not accurate. Okay, check it from every side that none of the molecule or the atom is outside of the grid. Grid will be not too small or too large. Its size is equal or probably equal to the size of the ligand. If ligand size is too large, then grid will be large. Okay, mean you have to arrange it in average, not too small, not too large. Okay, it is the most crucial step. If you practice it again and again, then you will be able to do it. At first it is difficult to learn, but yes. Okay, it's time consuming, but it is necessary for it. Now you can see that none of the molecules are outside of the grid. Okay, these are the confirmation or the dimension of the grid which you can write up in your research or thesis or research paper. Okay, minimize it and now simply click forward. Now docking is started. I select minimum atoms, not all which are you see in the 2D diagram, but you have to select all the residues with the same method for accurate results. Okay. If okay, the results are here. The results are not sorted here. Means the minimum energy is at the first. So we can arrange it in our Excel sheet. Simply download the Excel file. Now you can double click on the binding affinity column, then the large value or the best value will become up, but you can do the same thing in our Excel file also. Okay. Open the Excel sheet or result file you should download from the PyRx. And select the column B, which is the binding affinity column. Click sort. Select smallest to largest. Let's sort the column by giving small value molecule up, which is the best value of the docking. First is minus 10.9, minus 9.7, minus 9. Select the column C, and again sort smallest to largest. And sort. Okay. The values or the rows with RMS, JIB, UB with 0, we only need this. Okay. Otherwise, all the column below this will be discarded. We only need these rows. Simply click the first, go to the bottom, press shift and click the last row. Right click and select delete. Now we have only the result of the docking with RMST 0. Save your file. Click yes. Okay, it is the step of making a complex file. Open PyMOL. Go to your C drive, users, your desktop, and find MGL tools here. Click on PyRx, macromolecules. Click your receptor, and you will see the receptor and the ligands there. Drag your receptor file in the PyMOL. And the ligand file also, as we do in our previous lecture of blind docking. Okay, now you run. Now you can see that the ligand move in that grid. All the poses are in that specific grid. Ligand not move outside this area, which is our grid setting, grid conformation, grid dimensions. Save your file in the PDB format. Okay, I hope you will learn the site-specific docking with these easy steps. It's the complete video to perform. Now tell me anything in the comment section and ask any query. Like, follow, and share if you like the video. It is the step of checking poses. The exhaustiveness was 8 to 9, so there is now only 9 poses. The ligand is dancing. Please like, share, and subscribe.